Hello, I'm Dr. Mark Darcy, and I will now be giving a short lecture on photosynthesis. Let's get started. Photosynthesis is how a plant harnesses light energy to make chemical energy. So basically, um, light has a lot of energy trapped in the photons that are emitted from the sun. Plants take that energy. They also take water and carbon dioxide, so hydrogen and oxygen, and they form carbon-carbon bonds and carbon hydrogen bonds. So carbon bonds and carbon hydrogen bonds. This allows for the manufacture of sugars, but together with things like nitrogen, the plant can also, and phosphorus, can actually make amino acids, proteins, nucleic acids, um, fatty acids, all the building blocks of life. So pretty much all life on this planet depends on photosynthesis. Now, I'm not going to talk too much about the leaf structure, but obviously for photosynthesis to occur, a plant needs to take in carbon dioxide, which it does through the open stomata of the leaves. Stomata is basically Greek for mouth. These are little openings, usually on the underside of the leaf, surrounded by guard cells. The guard cells use potassium to become turgid. That means they can swell up to close the stomata, or they can deflate to open the stomata. This allows CO2 in, water to evaporate out, and also oxygen to leave the plant. In Greek, photos means light, synthesis means to put together. So we are converting light energy to chemical energy. All life on Earth depends on this process, more or less. There is some life in deep thermal vents that does not, but most life depends on this process. And all the oxygen on this planet is supplied by photosynthesis. Here is the general formula for photosynthesis. 6 CO2 plus 12 H2O plus light gives you 1 glucose, C6H12O6, plus 6 O2 plus 6 H2O. Carbon dioxide has 1 carbon and 2 oxygen atoms arranged OCO in double bonds in the molecule of carbon dioxide. Water has 2 hydrogen and 1 oxygen atoms arranged HOH in the water molecule. Photosynthetic reactions. The overall equation for photosynthesis is deceptively simple, but it's actually a complex set of reactions that requires 30 distinct proteins to take place. So we're going to go through some diagrams now and go through each step of both the light-dependent reaction and the light-independent reaction of photosynthesis. The light-dependent reaction is where you harness energy. The light-independent reaction is where you use that energy to make biological molecules. So here is a chloroplast. Chloroplasts have a double membrane. They have little discs called thylakoids, which is where the light-dependent reactions take place. There's also the stroma. This is the liquid of the chloroplast. This is where the light-independent reactions take place. Chloroplasts also contain their own DNA, which is circular, and their own ribosomes, which are 70S ribosomes, the same ribosomes you would find in bacteria, in prokaryotes, which hints at the origin of chloroplasts. So chlorophyll is found within the thylakoid membranes. Green light, which is not absorbed, makes chlorophyll appear green and is not used in photosynthesis. Chlorophyll absorbs red and blue light, and the energy from this is used in photosynthesis. So the photosynthetic pigments. Sunlight contains all the wavelengths of light. Most pigments, however, only absorb light at a certain wavelength. The primary pigment for photosynthesis is chlorophyll A. But there are accessory pigments, such as chlorophyll B, carotene, and xanthophyll. They give plants other colors than green. These trap energy from wavelengths of light that chlorophyll cannot. This helps the plant to become more efficient at absorbing light energy. The pigments are found in structures called photosystems. Here is a photosystem. Let us zoom in a little bit on that. So here is a photosystem. These are embedded in the thylakoid membranes. What you are seeing in this photosystem are a number of accessory pigments up the funnel of the photosystem. Light is absorbed by those accessory pigments and passed down to the reaction center. The reaction center is where the chlorophyll A is. Specifically, there's a pair of chlorophyll A molecules, either a pair of P700s or P680s. The 700 and 680 refers to the wavelengths of light absorbed in nanometers. In terms of light harvesting, photosystems are held by proteins in the thylakoid membranes. Accessory pigments collect photons of light, and the energy is transferred down the funnel or antenna to the primary pigment, the reaction center. The primary pigment is chlorophyll A. This is chlorophyll. Chlorophyll has what we call a porphyrin ring. This is the ring at the top here that contains magnesium. It's actually the magnesium that absorbs the sunlight. Chlorophyll A P680 absorbs light maximally at 680 nanometers and is in photosystem 2, which is in the thylakoid membranes, in the stacks of thylakoid. The stacks of thylakoids are known as grana, by the way. Chlorophyll A P700 is in photosystem 1 and has a peak absorption at 700 nanometers. Photosystem 1 is in what we call the intergranal lamella. They are the membranes that connect the different stacks of grana. There are also accessory pigments. For example, carotenoids absorb blue light to appear yellow or orange, carotene orange and xanthophyll yellow, and they pass energy to chlorophyll A. Photosynthesis has two stages. There are the light reactions and there are the dark reactions. The light reactions require light to occur. This involves the actual harnessing of light energy, and this occurs in or on the grana. The grana are the stacks of thylakoid membranes. The dark reactions do not need light to occur. This involves the creation of the carbohydrates. So products of the light reaction are used to form carbon-carbon covalent bonds in carbohydrates. This occurs in the stroma. So let's start with the light-dependent reactions. The inputs to photosynthesis are light, that goes into the light-dependent reaction, together with water. There are more inputs too, but we'll get to those. Oxygen is released as a waste product. But the main products of the light-dependent stage are passed on to the light-independent stage. This is also where CO2 is used to generate glucose, amongst other things. The inputs of a light-dependent reaction are light, water, oxidized NADP, but also ADP and inorganic phosphate. The outputs are ATP, oxygen, and reduced NADP. Reduced NADP is NADPH. So chlorophyll molecules are arranged in clusters called photosystems in the thylakoid membranes, as I mentioned. Each photosystem contains a pair of chlorophyll A molecules at its reaction center and will also contain chlorophyll B and other accessory pigments elsewhere in the photosystem. Photosystem 1 is found on the intergranal lamella, while photosystem 2 is found on the granal lamella. The process of making ATP from ADP, phosphate, and light is known as photophosphorylation. The process is using light energy to synthesize ATP from ADP and phosphate. It can be cyclic or non-cyclic and occurs when a high energy electron passes through an electron transport chain in a series of redox reactions in the thylakoid membranes. So we will start with cyclic photophosphorylation. Now, what we're looking at here is we are looking at photosystem 1. Specifically, what we're looking at is the porphyrin ring of photosystem 1. Even more specifically, we're looking at the electron in the outer orbit of the magnesium of photosystem 1. So light is absorbed by the accessory pigments and goes down the photosystem to that electron. So this only involves photosystem 1, results in the formation of ATP but not reduced NADP. Light is absorbed by the photosystem and the energy is passed on to chlorophyll A in the reaction center. This excites an electron enough for it to leave the chlorophyll molecule and enter an electron transport chain. As it moves down the chain, the energy released is used to synthesize ATP. Once the electron has lost all its energy, it returns back to photosystem 1. I will explain how the energy is actually used to make ATP shortly. But basically, the electron transport chain is embedded in the thylakoid membrane next to the photosystem, and that electron is passed down the electron transport chain in a series of redox reactions. Oxidation of one molecule, reduction of the next. The energy is used then to move H+ ions into the thylakoid space, and that will be used later to generate ATP. Non-cyclic photophosphorylation involves both photosystem 1 and photosystem 2. And produces ATP and reduced NADP. Electrons excited from photosystem 2, instead of returning to photosystem 2 at the end of the electron transport chain, replace lost electrons from photosystem 1. The electrons emitted from photosystem 1 are now used to reduce NADP rather than generate ATP. To reduce the NADP, you need H+ ions as well as the electrons. This is where water becomes involved. So let me summarize this slide before I move on to the next one. So if we look here, for example, at photosystem 1, light is absorbed by the electron in the chlorophyll A, in the magnesium of the chlorophyll A. This excites it so much that it leaves photosystem 1 and is passed down an electron transport chain embedded in the membrane. At the end of that electron transport chain, the electron joins with an H+ ion, a proton, to form hydrogen. And this is placed onto the oxidized NADP to reduce it and give you NADPH. This means an electron is now missing from photosystem 1 and needs to be replaced. It is replaced by an electron from photosystem 2. Light is absorbed by that electron in the chlorophyll A. It gets excited, leaves photosystem 2, and is passed down an electron transport chain. The energy released is used to synthesize ATP. Then, when the electron has lost its energy, it replaces the lost electron from photosystem 1. This means you have a lost electron in photosystem 2 that needs replacing. It's replaced using water. There's a process called photolysis, which uses light energy to split two H2Os into basically 4 H+ ions, 4 electrons, and an O2. The electrons are used to replace the lost electrons in photosystem 2 to keep the chain going. The H+ ions, they will be pumped into the thylakoid space, and that energy will be used to make ATP. And when the hydrogen ions are released from a thylakoid membrane back to the stroma, they will combine with H+ ions to make hydrogen and reduce the NADP. The oxygen will just be released as a waste product. Photolysis. Photosystem 2 contains an enzyme that splits water molecules when activated by light. The reaction is called photolysis. 2 H2Os gives you 4 H+ ions, 4 electrons, and an O2. The hydrogen ions are taken up by the NADP and combine with the electrons from the electron transport chain of photosystem 1. The electrons produced from photolysis replace the electrons originally lost from photosystem 2. So what about the H+ ions? Thylakoid membranes are impermeable to H+. So when you split the water, as the electrons are passed down the electron transport chain, the carriers transport H+ into the thylakoid space. A positive charge builds up, and an H plus and electrochemical gradient also builds up. Spanning the thylakoids are ATP synthase proteins. These allow the H plus ions to pass back from a thylakoid space to the stroma and synthesize ATP at the same time. Approximately 3 H pluses are needed for every ATP made. The H pluses are then available for the reduction of NADP. This is the chemiosmotic hypothesis. That is a light-dependent reaction. The inputs, we have water, we have ADP, we have phosphate, and we have oxidized NADP. The outputs are reduced NADP and ATP, which will be used in the light-independent reaction, and the waste product of O2, oxygen. So let's now explain what happens in the light-independent reaction, which occurs in the stroma. So it occurs via a cyclic pathway called the Calvin cycle, a result of a sequence of enzyme-controlled reactions requiring ATP and reduced NADP from the light-dependent stage, and also CO2. CO2 diffuses into the leaf through the open stomata towards the photosynthetic cells. In the stroma of the chloroplasts, CO2 is fixed by a 5-carbon compound called ribulose bisphosphate, RUBP, to form an unstable 6-carbon compound. So RUBP is a CO2 acceptor molecule. The process is catalyzed by an enzyme called ribisco, ribulose bisphosphate carboxylase. So this is a carboxylation reaction. Ribisco is made in the chloroplasts using chloroplast DNA and ribosomes. The 6-carbon compound, 3PG, immediately splits into 2 molecules of a 3-carbon compound called glycerate 3-phosphate, GP. This is where the ATP and reduced NADP from a light-dependent stage are used. They're used to reduce glycerate 3-phosphate, GP, to produce triose phosphate, TP, 3-carbon phosphorylated sugar. 1/6 of the TP is used to make glucose, amino acids, fatty acids, glycerol, or nucleic acids. 5/6 are used to regenerate more RUBP. This requires ATP to keep the cycle going. This is a kind of complex overview of the whole process, but here is a simplified version. So basically what is happening is ribisco, the enzyme, ribulose bisphosphate carboxylase, takes a 5-carbon RUBP and a CO2. They react together to make an unstable 6-carbon molecule called 3PG, which immediately splits into 2 GPs, glycerate 3-phosphates. You then use ATP and reduced NADP from the light-dependent reaction to turn the GPs into TPs, triose phosphates. 1/6 of the TPs are used to basically make everything. Sugars, fats, nucleic acids, everything. The other 5/6 are turned back into RUBP, which requires ATP from a light-dependent reaction, which keeps the cycle going. So the uses of the Calvin cycle products: GP can be used to synthesize fatty acids by entering the glycolytic pathway and being converted to acetyl coenzyme A. That would be explained in a respiration lecture. Triose phosphate is used to synthesize glycerol. GP and inorganic salts are used to synthesize amino acids. Pairs of TPs combine to form hexose sugars such as glucose. Glucose and fructose combine to form sucrose, the main translocatory sugar in plants in the phloem. Starch is synthesized in the stroma for energy storage. The structural carbohydrate cellulose is also synthesized. Okay, so that has been an overview of photosynthesis. I hope it was useful. Bye for now.